Welcome to Head and Neck Embryology Part 1. Today we'll introduce the pharyngeal arches, begin talking about the development of head and neck structures, as well as introduce some craniofacial abnormalities. I'm Dr. Catherine Moore, the histology wizard. The pharyngeal arches begin to develop early in the 4th week as neural crest cells are migrating into the future head and neck regions. The paired pharyngeal arches support the lateral walls of the primordial pharynx, which, as you recall from gut development, arises from the cranial foregut. The first pair of arches, which will give rise to much of the face, appear as surface elevations lateral to that developing pharynx, and the other arches arise as ridges on the future sides of the head and neck regions, with 4 arches visible at around 4 weeks. We'll begin by talking about the organization of the arches, called the pharyngeal apparatus. Here's a lateral view of an embryo, showing the 5 paired arches, 1, 2, 3, 4, and 6, named cranially to caudally. In humans, arch 5 is rudimentary, and 6 isn't seen at this stage shown here. We also know the first arch will separate into the maxillary and mandibular processes, or prominences, which we'll talk about later. The arches are separated by pharyngeal clefts, or grooves, that are also numbered in a craniocaudal sequence. If we slice this embryo and look inside, we can see the pharyngeal pouches which line the pharynx, and not shown here are the places where clefts and pouches meet, called the closing plates or membranes. Now let's take a closer look at these components. If we take an electron micrograph of an embryo and we section it and look inside, we can see the floor of the pharynx. In yellow, we see the endoderm lining the pouches, like most of the gut. In the middle lies the purple mesenchyme. This is the arch proper. And on the outside, we see the blue ectoderm forming the clefts. And the closing membranes are where the endoderm and ectoderm meet with no mesenchyme in between. Now let's go through this one more time, this time with a cartoon. Here we have that arch with its mesenchyme, which remember is early embryonic connective tissue, and that will give rise to musculature. We see the endoderm of the pouches, which give rise to epithelium and structures of the tongue and neck glands, and the ectoderm forming skin and neurogenic placodes. Later in development, much of the arch mesenchyme is formed from the migration of neural crest cells that, as you can see, form many of the face and neck structures. We'll talk about the pouches and the clefts in part 2 of this series, but first let's talk about the components of the arch itself. We'll check out what a typical arch looks like using this same cartoon. As neural crest cells migrate into the arches, the myogenic mesoderm will also move in, forming a core muscle primordium that gives rise to head and neck muscles. Lateral plate mesoderm and invasive angioblasts will migrate in and form the aortic arches. A cartilaginous rod will form the skeleton of the arch, and finally, sensory and motor nerves will supply the mucosa and muscles from each arch, and these are derived from neuroectoderm of the primordial brain. All of these tissues together contribute extensively to the formation of the face, the nasal cavities, the mouth, the larynx, the pharynx, and the neck. Each arch will contribute different components. So now let's talk about the derivatives for each arch individually. Keep in mind that the related structures are developing at the same time, and this is important because it means that genetic lesions that affect a particular arch affect multiple developmentally related structures. For each aortic arch, an artery arises from the truncus arteriosus of the primordial heart and passes around the pharynx to enter the dorsal aorta. Much of the aortic arch from arches 1 and 2 disappears, leaving just a few adult structures. Now aortic arches 3 and 4 and 6 will remodel, and they will give rise to the common and internal carotids, the aortic, subclavian, and pulmonary arteries. Now what about the cartilaginous components? Well, this cartoon outlines the major derivatives of that cartilaginous rod, and they're color-coded for each arch. Recall that bone derived from cartilage does so via endochondral ossification, and other facial bones will also develop around that cartilage by intermembranous ossification. Arch 1 cartilage, also called Meckel's cartilage, will give rise to the malleus and incus bones of the middle ear, some ligaments, and it not only gives rise to parts of the mandible bone, but also guides the intermembranous ossification that will give rise to the rest of the arch 1 bones, seen here in orange. Arch 2, also called Reichert's cartilage, will give rise to the stapes of the middle ear, the styloid process of the temporal bone, the lesser horn of the hyoid bone, and some ligaments, while Arch 3 cartilage will give rise to the greater horn and part of the body of the hyoid, as well as some ligaments. Arch 4 will form some thyroid cartilage, and Arch 6 will form the cricoid cartilage. Now the epiglottis forms from mesenchyme, and the tracheal rings themselves form from splanchnic mesoderm. Speaking of mesoderm, the mesoderm of the pharyngeal arches makes major contributions to the muscles of the face, as you can see here from this color-coded cartoon. Now normally I would say you don't need to know all of these, but the arch derivatives are board's favorites. So let's walk through these together. Again, let's start with Arch 1, which forms the muscles of mastication, the temporalis, the mylohyoid, and the masseter, along with other muscles. Arch 2 gives rise to the muscles of facial expression, and boy are there a lot of them, including the frontalis, the orbicularis oculi, and the buccinators, as well as the stapedius, the stylohyoid, and the auricular muscles. In contrast, Arch 3 is pretty easy, since it just forms the stylopharyngeus. The fourth arch forms the cricothyroid, the levator palatine, and the pharyngeal constrictors, and Arch 6 forms the laryngeal muscles. Now last but not least, let's talk about the nerves. Each arch essentially has its own cranial nerve. The visceral afferent nerves supply the muscles derived from the arches, while the dermis and mucous membrane areas come from special visceral afferent nerves. The facial skin is supplied by cranial nerve 5, the trigeminal nerve, but only the maxillary and mandibular branches are going to supply those first arch derivatives. Remember that the first arch divides into maxillary and mandibular arches. And this includes the muscles of mastication and face, the teeth, and the mucous membranes of the nasal cavities, the mouth, the palate, and the tongue on the sensory side. Now the facial nerve, cranial nerve 7, supplies the second arch. Cranial nerve 9 supplies the third arch, while the superior laryngeal branch and recurrent laryngeal branch of cranial nerve 10, the vagus nerve, supply Arch 4. Now I realize there are a lot of derivatives that I've talked about and a lot of derivatives to learn, so I highly suggest finding a table that just summarizes all these derivatives and learning them, and there are a lot of good tables out there. Now I'd like to go back and touch on the importance of the neural crest and the control of migration into the arches. The homeobox gene family, or Hoxgenes, are critical regulators of embryonic development, and we've seen their influence over and over again as we discuss the different body systems. The Hoxgenes are major controllers of anterior-posterior patterning in the body, and the arches illustrate this relationship beautifully. In this cartoon, you can see the hindbrain with color-coded expression of Hoxgenes in each rhombomere, cranially to caudally. Now, while the specific genes aren't that critical to learn, what's important is that neural crest cells expressing the specific Hoxgenes will migrate to locations in the pharyngeal arches, and they carry that anterior-posterior patterning from the Hoxgenes with them. So each arch then has a Hoxgene code. This cartoon shows the different codes of each arch, so that, for example, here, Arch 1 doesn't express any Hoxgenes in this family, but Arch 4 expresses all of them. Now, what happens to these arches when we eliminate the expression of all these Hoxgenes? If you guessed that the arches will develop all as Arch 1, you've guessed correctly. Now, why is this even important? Well, we know that mutations in some Hoxgene family members can affect the structures of the face and neck, and this results in duplications or deletions of critical structures. Now, I'd like to just touch on the relationships of the muscles and nerves of the pharyngeal arches. So this is a fairly complicated figure, but it really shows the mostly straightforward relationships between the neural crest migrations, the nerves, the muscles, and pouches of the pharyngeal arches. And this actually helps us think about congenital abnormalities. Many times, the muscles and nerves originate at the same level, such as we see here with arches 2 and 3. But sometimes the relationship can be more complicated. For example, the muscles of the tongue do not arise from the arches. Those progenitors travel in from the somites, but they're innervated by the cranial nerves in order. Now we're going to talk about some problems that arise from aberrant arch development. But before we do that, we have to talk briefly about the development of the face. So facial development actually depends upon 5 processes or prominences, and these are simply swellings of the tissue. And in this electron micrograph, you can see the swellings. And in the matching color-coded cartoon, you can see the frontonasal prominence in pink, the paired maxillary prominences in purple, and the mandibular prominence in blue. Now, you may have only counted to 4, but you didn't count wrong because the mandibular process initially appears as 2 bulges, so it counts as 2 processes, hence 5. Here you can see some cartoons showing facial development at 4 weeks, 6 weeks, and in the adult, and I've color-coded them. So most of this growth and change is going to depend upon expansion of neural crest populations. We have the blue mandibular process that will form the bottom of the face, including some of the tongue and chin, while the purple maxillary processes will give rise to that mid-face, the upper lips, the palate, etc. And that frontal nasal process in pink forms the forehead and eventually part of the nose. So to review, here are some different cartoons of the developing face showing the frontal prominence in pink, the paired median nasal prominences in green, the lateral nasal prominences in orange. These actually both arise from that frontal nasal process, and the mandibular and maxillary processes. Disruption of development of any of these processes or prominences can result in a variety of defects, which we'll look at in a few minutes. Here in this electron micrograph, you can see all of the processes, but what you can also see is they aren't quite connected. There are actually seams or clefts between them. And these prominences were going to have to grow together and essentially fuse together or merge together, kind of like smoothing out the seams, if you will. So let's take the mandibular process here, which has these 2 bulges sort of like a dumbbell. But what you can note is that the epithelium is actually all connected, as if you put a couple of balls inside a sock. Now we know that neural crest cells are going to migrate in and proliferate, and that proliferation or filling in process will cause those indentations of the dumbbell to smooth out until the end result is one mandibular process that has these nice smooth edges. Now I've shown you this process for the mandibular process or prominence, but any place that these clefts reside or these processes meet, such as the median nasal processes next to the maxillary processes in purple, you see the same kind of mechanism. Now, of course, you can imagine some major issues with this whole Mergence process. The neural crest can fail to migrate in, it can fail to proliferate, and overall the merging can fail. So what would that actually look like? Again, let's take that mandibular process. Here's a few examples. You may actually recognize some of these people. And each of these images is showing a defect in facial merging, leading to a minor cleft chin in someone like Ben Affleck to much, much more severe defects. So let's look at a few specific examples of failure of merging. Here are 2 examples of cleft lip, the most common facial cleft. And this arises from a failure of the median nasal process to fuse with the maxillary process. And this can be uni or bilateral, with or without cleft palate, as we'll talk about in part 2, but it can lead to many, many different problems. Other clefts can occur, such as oblique facial cleft, and this arises from the failure of the maxillary and lateral nasal prominences to merge, as seen in these cartoon examples, and these can be quite severe. Another example is median cleft lip. Now, what processes do you predict would fail to fuse here? If you said the median nasal processes, you're absolutely right. Now, there are many different combinations, and I've just shown a few here. Most of these can be repaired surgically, and many occur in combination with other facial or body defects. Now, another classical congenital problem that involves the face that I'd like to discuss is first arch syndrome. Here, neural crest cells fail to migrate properly into the first arch. This can either be due to inadequate proliferation and/or actual migration defects, and in some cases results in a syndrome called Treacher-Collins syndrome. Treacher-Collins syndrome results in very characteristic phenotypes, including hypoplasia of the jaw and zygomatic bones, micrognathia, coloboma of the eyelids, and many more, including tongue, palate, and airway issues. And all of these phenotypes can be traced back to problems with Arch 1 development. Another Arch 1 disorder is Pierre Robin sequence, where one defect, mandibular hypoplasia, leads to the tongue being misplaced. And since we know the tongue has to move down during palate formation, this leads to cleft palate. And cleft palate can often lead to airway issues. And in these images, you can see that this infant has a tracheal tube to help with breathing. Now, if you've forgotten what a syndrome or a sequence is, you should go back and check out my earlier video. Facial abnormalities can also be caused by in utero exposures, such as vitamin A. Maternal use of Accutane used to treat acne can cause fetal retinoid syndrome. These patients have craniofacial defects, and because neural crest is affected, cardiac outflow tract and limb defects are often comorbid. Another environmental cause of facial deformities is fetal alcohol syndrome, which represses neuroectoderm and neural crest induction, leading to facial abnormalities seen here. Finally, let's end with some congenital malformations arising from aberrant development of the skull, also known as craniosynostosis. In order for the brain to grow, the skull contains sutures that contain progenitor cells for bone, and this allows for rapid bone formation. Now, eventually, these sutures will fuse. However, sometimes one site will close too early, and this will cause increased growth at other sites. And this can result in deformation of the brain and the skull. And here are some examples. Now, we know that there are a number of different syndromes that are associated with craniosynostosis, and some of those are shown here. Now, what causes this problem? We need to think about proliferation and differentiation as a teeter-totter. It's a delicate balance. And we know that proliferation of bone cells in these sutures is controlled in part by FGF signaling. And activating mutations in the FGF receptors leads to increased FGF signaling, and that tilts the teeter-totter, as you see here, towards differentiation, so towards premature suture fusion, and that can result in the deformations of the skull that are seen here. We also know that neural crest is crucial for skull bone development, and so imbalances again in neural crest can lead to craniofacial abnormalities. So today we've covered the basics of the pharyngeal arches. We've talked about their derivatives and discussed some malformations associated with those arches. In part 2, I'll cover the pharyngeal pouches and clefts and their derivatives, as well as discuss the development of the tongue and neck structures. Please check out that video. Please like and comment. And thanks again for stopping by.